Hello, today we're taking a closer look at our cell tracking reagent DIV000F1. Designed for researchers like you, this reagent simplifies the process of tracking cell internalization. Let's dive in. Harness the power of fluorescent lipid nanoemulsions. This reagent is essential for tracking cell internalization, offering a reliable positive control. Whether comparing with other delivery systems or exosomes, expect consistent results. Inside this reagent, you'll discover two crucial vials, DIV000F1 and DIVTech. We've also included a pair of 1 milliliter micropipette tips for precision. Now, let's get down to the protocol. Start by adding 100 microliters of ethanol to the DIV000F1 vial. Gently mix by pipetting up and down. This ensures a smooth and consistent solution. Next up, the DIVTech vial. Introduce 900 microliters of ultra-pure water. PBS or HEPAs can be alternatives if needed. For more buffer suggestions, take a look at Table 1. Time to combine. Swiftly transfer the DIV000F1 content into the DIVTech vial. Ensure you're using the same micropipette set to maximum volume and tip. A firm and quick action is key for optimal mixing. For customized and optimized prototypes, you can contact us at info@diversatechnologies.com.